So, hello everyone, I am Julia MUñOZ-GUARINOS and I'm doing a PhD at the Laboratory of Human Evolution here at the University of Burgos. And today I'll be presenting a talk on the impact of senescence and menopause on femoral cross-sectional geometry. But first, let me introduce this topic from different perspectives. Menopause entails diverse social and cultural implications that vary based on the norms and expectations of each society. In some places, pressure to maintain youthfulness can impact perceptions of beauty and femininity, while in other contexts, rituals and celebrations honor this biological transition. Nevertheless, our understanding of how menopause was conceptualized in past population remains unclear. And to explore this social construct in the past, the initial step involves identifying it in the archaeological record through the analysis of skeletal remains. All social constructions are shaped by biological facts, and menopause is no exception. In our life history model, which is represented in this figure, menopause marks the conclusion of our fertile period at around 50 years old. While other mammal species may undergo menopause if they live long enough, our species is one of the few, along with orcas, pilot whales, and narwhals, that have a post-reproductive period as lengthy as the reproductive one. After menopause, it is important to pay attention to bone health, since hormones influence the bone modeling process during growth and continue to regulate bone modeling in adulthood. Aging is characterized by skeletal changes, which manifest differently in cortical and trabecular bone and in women. These changes are tied to hormonal fluctuations during menopause, where estrogen levels decrease, disrupting the balance between bone deposition and bone resorption. Numerous studies have been conducted in the current population regarding bone loss after menopause, but in archaeological samples, we have no information on this matter. Thus, the main aim of our study is to evaluate the effects of senescence and menopause in an archaeological sample by examining femoral cross-sectional geometry. To do that, we have analyzed 39 femora from adult individuals from the medieval population of San Pablo, host here at the Laboratory of Human Evolution. Our sample comprises 22 females and 17 males. Sex was estimated following FENIS, and age was estimated using Transition Analysis 3 software program. Then the sample was categorized into 3 groups that you can see here: males, females under 50, and females over 50, as the mean age of menopause in living population is 50 years. Then we extracted the femoral cross-sections at various locations along the bone using CT images and visualized them with the MIMIC software. The variables under analysis can be categorized into 2 types: the area variables and the shape variables. Area variables, which you can see here in the left, encompass total area, cortical area, and the medullary cavity area, while shape variables involves moments of area of the anatomical axis, as well as maximum and minimum moments of area. As Raf proposed, to identify significant differences in bone structure, it is necessary to control for the effects of body size. For this reason, the geometrical properties were analyzed and standardized by estimated body mass. And body mass was estimated using the vertical diameter of the femoral head and the regression equations developed by Raf and colleagues in 2012 for adult individuals. Then these geometrical properties were regressed on estimated body mass using a linear regression that you can see here in the right, and standardized residuals, which are the raw residuals divided by the standard deviation of these residuals, were then used in comparison. Finally, Mann-Whitney U-tests were conducted to identify significant differences among the groups in which have divided our sample. So due to the extensive amount of analysis conducted, only the most significant result will be presented here. Starting at the 20% sexual level of the diaphysis, we find significant differences in the total area between women over and under 50 years. At the next section level, at the 35%, we find these differences not only in the total area, but also in the medullary cavity area. Then, in the mid-shaft section and in the proximal third of the diaphysis, the differences between women over and under 50 are observed in the cortical area, but not in the previous variables. Moreover, we also observe significant differences at midshaft in the ratio of moments of inertia of the anatomical axis, being the median of women over 50 significantly higher than women and than other women and men. Finally, at the femoral mid-neck cross-section, we observe the same trend as in the distal third of the diaphysis, where the only difference between women is found in the total area. The typical course of bone modeling and remodeling in women follows a distinct pattern. During puberty, women exhibit a greater emphasis on endocortical rather than periosteal bone apposition, resulting in more gracile bones compared to men. In the initial stage of bone loss following menopause, the marked deficiency of estrogen significantly accelerates this endocortical resorption and diminish periosteal apposition, leading to rapid cortical thinning. The impact on trabecular bone loss during this phase is even more pronounced, and in a subsequent phase, endocortical resorption persists at a slower pace, and cortical porosity develops, making the bone more susceptible to fragility fractures. However, we are observing two distinct patterns along the diaphysis and the femoral neck. In our sample, in the distal third and in the femoral neck, we observe a difference in the total area. So the most notable trend is the decrease in periosteal apposition, while in the proximal third, we observe differences in cortical area. This suggests that in our sample, endocortical resorption accelerates more in these regions of the diaphysis than in others. Moreover, at midshaft, we observe significant changes in women over 50 related to geometrical compensatory changes in the moments of area of the anatomical axis. Previous studies have shown that, unlike men, women do not show geometrical structure change that would compensate bone loss in the femoral midneck. However, our results suggest that this compensation do occur at midshaft in women over 50. In this figure, we can observe some of the most representative individuals within the groups into which we have segmented our sample, being evident that medullary cavity expansion due to the acceleration of endocortical resorption, for example, at 65 or 80%, it is more evident. But we are aware that bone loss is influenced by a combination of genetic and environmental factors, and the interaction between them is really complex. Genetics influence bone loss through various mechanisms, including the determination of the peak bone mass, the regulation of bone modeling process, the hormonal balance, the vitamin D metabolism, and the formation of crucial bone proteins like collagen. On the other hand, the environment we live in has a profound impact on the strength and resilience of our bones. Adequate nutrition, particularly sufficient intake of calcium and vitamin D, is crucial for bone formation and mineralization. And we must consider also the importance of physical activity. Regular weight-bearing exercise, such as resistance or impact activities, played a pivotal role in maintaining bone density by subjecting bones to mechanical loading. And finally, sunlight, often underestimated, is a natural source of vitamin D, which is vital for the synthesis of this essential vitamin, contributing significantly to calcium absorption and consequently robust bone health. In summary, interpreting the variation in cross-sectional variables in relation to senescence and menopause presents a challenge. Nevertheless, our findings suggest that menopause status exerts an impact on all the variables and cross-sectional areas analyzed in these studies, showing thinner cortices and compensatory geometrical changes in women over 50 years. Furthermore, these changes can be discerned in archaeological populations, and this study raises the possibility of using changes in cross-sectional geometry of the femur as an indicator of postmenopause, a topic we will continue to explore in future research. Finally, we would like to thank our colleagues from the Laboratory of Human Evolution. Thanks also to the Spanish government for their financial support, and to the government of Castilla y León and the European Union for the PhD grant from which I benefit. Finally, of course, thank also to the organization of this Congress for accepting our proposal, and thank you all for the attention. We would be very pleased to hear your feedback. Thank you.